dancing cheek to cheek. Oh, I love to climb a mountain and to reach the highest peak, but it doesn't thrill me half as much as dancing.
All right, good afternoon. I hope everybody had a fabulous lunch. My name is Don McMorrin. I'm the Extension faculty member at WSU Skagit County Extension, and it's my great pleasure to be able to introduce our next speaker. His name is Tobin Peever. Tobin is an associate professor at WSU Pullman. He has a teaching assignment as well as a research assignment, and he also splits time between Pullman and WSU Northwest Rec in Mount Vernon, where he has an assistant grad student that is working on projects there as well. So it's my great pleasure to introduce Tobin. If you'd please give him a round of applause. Thank you.
Thank you. Can you hear me okay? How do I move this in a little bit? How's that? Can you hear me okay? Doesn't sound like it. I might have to hang on to it. Yeah. So this is the pointer here, which is this one. Can you hear me now? That's better. Okay. Speak a little louder. How's that? Okay. You got to turn this one off. How's that? Is that better? Let's go. Okay. Okay. Thanks very much to the organizers for giving me this opportunity to talk about some of our work with fungicide sensitivity of Botrytis scenario. Okay. I don't know. Is that better? A little bit higher. It seems to be sagging a bit. So this work, it's pretty labor intensive work and involved a lot of people. I've listed some of the people up here. In the lab in Pullman, David Dutton is a technician who's been doing a lot of these assays that I'll show you. Olga Kojar is a PhD student. She's in Pullman, alternates between Pullman and Mount Vernon. She's been involved in a lot of the sampling that for this project, as well as some additional, some other projects that she's working on. Delphi Hardeveld is a postdoc. I think many of you know her. She's based at the Mount Vernon station. We have a couple of undergraduates, Adrian Alcocer, who's was a student at University of Idaho. He's working in the Pullman lab. He's done a lot of these assays. And Emreen Tour is an undergraduate from Western Washington University. She's been working in the Mount Vernon lab. So lots of people involved. Funding for this project comes from the Washington Red Raspberry Commission, the Washington Blueberry Commission, from the Washington State Commission on Pesticide Registration, and we recently got a grant to continue this work it's a Washington State Specialty Crop Block Grant, so we'll be able to continue some of this work for a few more years, which I think is going to be really good. So with that, let's see if I can advance this. So the objectives were to estimate sensitivities to the 5 major fungicides that are used to control Botrytis cinerea in berries in Washington. And this is a follow-up study, a study initiated by Alan Schreiber, Steve Midbow, Chris Clemens, and Jim Ataskavich in 2012 and 2013. So we followed up the initial, when I got involved in the project, our first samples were from 2014. I presented some of that work last year. And then we sampled again in 2015. So we're really on the second year of this project since my involvement. So the fungicides that we're interested in are listed here. Fenhexamide, so this is the active ingredient in Elevate. I'm going to be using the chemical names for these. So Elevate, you're familiar with Elevate. Fenhexamide this is a sterile biosynthesis inhibitor. It's in FRAC group 17. Rovral, iprodione, this is an osmoregulation inhibitor. It's in FRAC group 2. Switch, this is a mixture of 2 fungicides of saprodinil, which is a methionine biosynthesis inhibitor, FRAC group 9, and fludioxonil. which is another osmoregulation inhibitor, different mode of action than iprodione, in frat group 12. So we're separating the two components of that, of switch, of the mixture, and evaluating the sensitivity to each of those components independently. And then another mixture, pristine, boscalid is the component of that mixture that's effective against botrytis. So we've been focused just on that component of the mixture. There's also a strobilurin component of that we haven't looked at. This is a respiration inhibitor, and it's in frat group 7. So those are the 5 fungicides we've been focused on evaluating the sensitivity to. So the methods, they're fairly straightforward. We basically evaluate the sensitivity of isolates on a solid medium in a Petri dish. So we amend that medium with different concentrations of the fungicide. We measure the growth of those fungal isolates, and we measure the growth relative to a control where there's no fungicide present. So all of the data that I'll show you is expressed as a % of the control or proportion of the control. So it's how well the isolates grow on the fungicide amended medium relative to its natural growth rate in the absence of the fungicide. So the data I'll show you is this % of control or proportion of control. So this is an estimate of how sensitive these isolates are to these different fungicides. Okay. So this is what the assay looks like. So you can see we typically evaluate 4 isolates, 4 individuals per plate. So you can see 4 individuals start, they grow towards each other in this plate, and then we can measure how far they grow after a period of time. So this is no fungicide here. This is a plate containing one of these fungicides here. And what you can see is that in some cases, so the orientation is the same. So these 2 individuals here are here. So in some cases, these are not affected by the fungicide. So they're quite insensitive to the fungicide. There's no real apparent growth difference between them here. In this case, there's a slight inhibition of this particular isolate, so the growth is slowed down slightly here, versus a strong inhibition of growth for this isolate. So this isolate here, same position of the plate, is much more sensitive to that fungicide. Okay, so all of the data I'll show you is basically taken from this kind of assay where we're measuring the growth rate at a set period of time relative to how well it grows on the control plate. So if you plot that out, this is what it looks like. So this is a fenhexamide growth curve. Elevate fenhexamide growth curve. This is from some raspberry samples in 2014. We have % growth of the control over here, okay, versus the concentrations here. So this is the growth at 0 or the controls. This is 0.5 parts per million. This is 5 parts per million, so increasing concentration as you go from left to right. And what you can see is there's basically 2 families of curves here. There's this set up here. Okay, so each colored line is an individual isolate of the fungus. So the response of these individuals is these are quite less sensitive. They can continue to grow quite well at 5.5 parts per million and even at 5 parts per million, versus this family of isolates here, much more sensitive. Okay, and then we can classify those isolates as sensitive or as less sensitive to the fungicide. And in most cases, this is what we see, this kind of bimodal distribution where you have 2 types. To most of the fungicides that we look at, we can classify them into 2 or sometimes 3 different types. Okay, so our sampling strategy, as I said, this was part of a larger project. My graduate student, Olga Kojar, is interested in looking at the host specificity of Botrytis cinerea on different berry crops. She's interested in gene flow, fungicide resistances that we'll talk about here. So she did a lot of sampling this summer, and there were some of you in the audience participated in that by allowing us to sample your fields, and we thank you for your cooperation in doing that. But we sampled in 2014, when we first started this, we sampled primarily from raspberry. We had a few samples from blueberry, and that was about it. We sampled some wild blackberries. But this year and this past year, we had a much more extensive sampling. In total, we've got about 1,000 isolates or so. And I'll show you some of the strategies for some of the other projects, but we were sampled from many different hosts this year. So we sampled from blueberry, raspberry, strawberry, blackberry, wild blackberries, and also a field of currants. And we sampled in different geographic regions. So we sampled in Skagit County, Whatcom County, and also from Island County. The other thing, we tried to focus our sampling on fields that were managed in different ways. So we focused on fields that were managed in a conventional way using high fungicide inputs, substantial fungicide inputs. We sampled from organic fields. We also sampled from, in some cases, from fields. I didn't list it here. I should have. From what we call low input. So it's known that the fungicide inputs into that system were minimal. And then we also sampled from, in some cases, for some of the crops, from fields that had not been treated for a number of years because they've been abandoned from production. Okay, this was true of the strawberries, some of the strawberries that we sampled. So we had a whole series of different management regimes from the different fields that we were able to sample. And this table shows you the number of fields that we sampled and the number of isolates that we obtained from the different crops and fields. Okay, so blueberry. So our sampling strategy here, we sampled 3 blueberry fields in Skagit. All of the tables that I'll show you with samples, how we sampled, I'm going to use the same color codes. So we have the Skagit fields here in blue, the Island County field in red, the Whatcom fields in black. And here this shows the different kinds of management regimes that were used, so conventional, low input, so forth. So we sampled 3 fields in Skagit, 1 in Island, and 4 in Whatcom for blueberries. Okay, so what do we find? So I'll show you a series of these slides as well. So this summarizes all of the responses so we were sampled we would examine about 15 isolates, 10 to 15 isolates per field that we sampled. And so these are the frequencies of insensitive isolates. So we categorize these as sensitive or insensitive. These are the proportion of insensitive isolates in that particular field. Okay, so we can look down, for example, for fenhexamide, you can see the frequency here is about 20%. And it varies significantly from field to field. If you look down here, you can see some fields where we didn't detect any insensitive individuals at all, up to 70% and everything in between. What we found across all of the samples, every isolate that we've sampled from Washington, this is true also for British Columbia, we have not found highly insensitive isolates to either iprodione or fludioxonil. Okay, we've not found highly insensitive isolates to either of those fungicides. And you can see that here. But all of the samples that I show you from the different crops, you'll have the same pattern. We're not seeing highly insensitive isolates to either of those fungicides. You look down here, boscalid. So we have, again, a lot of variation from field to field. So you see here about 30% up to 100% and different frequencies across different fields. Okay. So we're seeing insensitive isolates to fenhexamide, to boscalid, to cyprodenil, not to fludioxonil, not to iprodione. So if we overlay the management on here, so these are 2 fields that were low input. Okay, we classified as low input, not receiving a lot of fungicide input. And what you can see is they generally have much lower frequencies of insensitive isolates to all of the fungicides. So there's a strong correlation between the amount of fungicides that's being used and the frequency of insensitive isolates that we're sampling, which makes sense. There's a lot less selection in these fields for insensitivity to the fungicides. And we've seen this pattern over and over again. I'll show you as we go through. It's the same pattern in these low input fields, in organic fields, in these wild blackberry populations, places that have received low sprays or no sprays. We're seeing low frequencies of insensitive isolates. It's a very straightforward message that the more you use these fungicides, the more we'll see the populations respond, and you see higher frequencies of insensitivity. This is an interesting field here. For fenheximide, there's a report in the literature of fitness costs associated with fenheximide resistance. And what that means is that in the absence of the fungicide, isolates that are insensitive to fenheximide are not as fit as the sensitive isolates. That's the absence of the fungicide. And so the prediction from that is that if once you withdraw use of that particular fungicide, that the resistance, or the insensitive population should decline. And this is what we think we're seeing with iprodione. There was control failures with iprodione in this region 15 to 20 years ago. We no longer see highly insensitive isolates to iprodione. Okay. The same thing may be happening with fenheximide. I'm going to make it's very speculative at this point, but we are seeing a few hints that the frequencies of fenheximide may be on the decline because it's no longer been used. It's been a couple of years now, it hasn't been used. And this is an interesting field here. We didn't sample it previously, so we don't know what the fenheximide insensitivity frequency was in that field. But if it's a typical Whatcom field, it would have been about 60 or 70%, because that's the average in Whatcom that we see for fenheximide. But look at this. We could not see an insensitive isolate in that field in 2015. The last spray of fenheximide in that field was in July of 2012. So, and there's some other data that I'm not going to show you, but there's hints that fenheximide, the frequency of insensitive isolates may be declining. So we need to follow some of these fields where we have high frequencies of insensitivity of fenheximide through time, and I think we'll be able to do that with the new block grant to see what happens to fenheximide's insensitivity over time. Okay, this is just a different way of looking at the data. So what I've done here is to color code. So fenheximide is going to be in teal, boscalid in red, cyprodenil in green. So we're not going to be focused on fludioxonil and iprodione since we're not seeing insensitive strains. We'll just focus on these three. These are the fields across here. I've just numbered these. These are the counties where these fields are located. And what I've done is to the solid portion of the bars is the frequency of sensitive isolates. Okay, we're half time. The hatched portion is the insensitive isolates. So you can see and they all add up to 1, so you can see what proportion of each type are in these different fields. And what's striking is you can see immediately the low input fields are right here that have higher, there's more of the solid bars, so higher frequencies of sensitive strains relative to others. But what's also really interesting when you look at this is the really large differences in frequencies among fields, okay? So one of the questions we're interested in with Botrytis is Botrytis this large population that affects lots of different berry crops? Is it the same on different berry crops, or is it different? Is it specialized on different berry crops? How much does it move between berry crops? How much does fungicide insensitivity that you might select for in blueberry affect raspberry field next door and so forth? So how much movement how much is the pathogen moving around? How much are these insensitivities moving around? From this data, it looks like these populations are quite local. Okay, so meaning that the fields, there's a lot of variation from field to field, so the management within the field seems to be the important driver of these frequencies that we see of insensitivity, which I think is really interesting. So what about raspberry? So I said we sampled raspberry in 2014. So this is 2015. So these are the samples from Skagit, same color code. These are the Whatcom samples here. We had a low input field in Skagit. The rest were conventional. You see a similar sort of pattern, a lot of variation from field to field. So for here example, for fenheximide, no insensitive isolates, 40%, 70%, and so forth, 100% here. Okay, same story for fludioxonil iprodione, no insensitive isolates, and a lot of variation for boscalid and cyprodenil. What's interesting about the 2015 sample, and we're sampling different fields in 2015 than we did in 2014, but we seem to have much higher frequencies of insensitivity to boscalid and saprodinil. Fenhexamide is about the same that we saw in 2014, but for boscalid and saprodinil, the frequencies appear to be much higher. Now, I don't know if that's because there's a lot of variation from year to year, or we're sampling different fields, or if the withdrawal of the use of Elevate has made us more reliant on, and we're using more boscalid and more saprodinil, more switch, and we've actually selected for more insensitive strains. But it's markedly different from 2014 to 2015 for those two fungicides. Okay, this is our low input field. So again, you can see this correlation between the amount of fungicide going in and insensitive isolates. Another way of looking at it, I'm running short on time, so I'll move ahead here. Strawberry samples, same idea. We sampled 4 in Skagit, 1 field in Island, and 3 in Whatcom, listed here. Here we had quite an array of different management regimes. So we had conventional fields. We had a couple of untreated fields. So these were basically fields that had been abandoned from production, but we could still go in and sample the fruit. We could still go sample Botrytis. Low input, and also an organic field in Skagit we were able to sample. So a wider array of different management schemes. If you look at this, so same pattern for fludioxylin and iprodione. Again, a lot of variation field to field. In general, what we see are lower, if you look down here, here, and here, much lower frequencies of insensitive isolates that we sampled from strawberry compared to the other berry crops. And I'll show you a summary of that at the end. If we overlay, this is the organic field. Okay. No detected insensitive isolates for any of the fungicides. Low input. And the untreated fields. So again, a strong correlation between fungicide use and the pattern of insensitivity that we see. So again, you can see if you look down here, these low input fields, they pop out at you because you have the bars, predominantly the solid colors. Okay, so low frequencies of insensitive strains in those fields, but still a substantial amount of field-to-field variation. That's a common theme that we see with all of these samples as we go through. Okay, so how much time have I got left? 10 minutes or so? 10, okay. So if we compare across if we put all this data together and compare them side by side, so this is a mean frequency for the 3 fungicides that we're focused on, fenhexamide, boscalid, and saprodinil, same color schemes here. This is the proportion of insensitive isolates. So this is our raspberry 2014 sample here. So I mentioned we see about 60 to 70% on average insensitive isolates to fenhexamide, to Elevate. This was the about 15 or 20% insensitivity to boscalid and to saprodinil in 2014. This went way up in 2015. So slight decline here. This could be due to because we sampled different types of fields. It looks like in Whatcom, really the frequency is about the same, about 60%, but much higher frequencies of insensitivity to boscalid and saprodinil in 2015. I don't know why that is exactly. I think with subsequent sampling through years, we'll be able to see if we do see a lot of turnover, a lot of variation from year to year, or if this is some kind of trend that there was more selection recently for those insensitivity to those particular fungicides. Strawberry much lower, as I mentioned, so the sensitivities to all 3 are much lower on average compared to the other crops. Blackberry, I didn't show you blackberry data. We sampled from several fields of blackberries in Skagit and Whatcom as well. Some are intermediate, and then the blueberry results are here. Okay, so that's on average. So these really conceal the substantial variation field to field that I showed you in the other graphs. But on average, we do have a substantial amount. There's a substantial level of insensitivity to all 3 of these fungicides. More to fenhexamide. The fenhexamide insensitivity is concentrated in raspberry, but there's a lot of insensitivities to these other fungicides, and it's across several different berry crops. Okay, so part of the sampling strategy that Olga pursued this year, she wanted to identify fields of berries that were located very close to each other, so adjacent crops of different berries. And the idea behind it is she samples from these different fields and is interested in how much the pathogen's moving between those berry crops on a very small scale. So they're as close together as we can get. How much are they moving between those two? So it's a good opportunity. We took some of those samples, looked at frequencies of insensitivity, and what you see, for example, this is a paired site here where we've got strawberry in this kind of pink-red color, blueberry in blue, blackberry in this dark blue. What you can see is in some cases, so this is fenhexamide, boscalid, and saprodinil sensitivity, again, proportion of isolates here. Some cases, they're pretty similar across the different crops, but in some cases, they're really quite different, okay? Down here, they look more similar, so this is a paired site where we have strawberry and blackberry. Generally low levels of insensitivity and fairly similar across the two crops. Down here, this value is not missing here. This is 0. So for example, for boscalid, this is a paired raspberry and blackberry field. We have 100% insensitivity to boscalid in blackberry and 0 in raspberry, even though those two crops are right beside each other. So again, it reinforces to me this idea that Botrytis populations are probably quite local. And we're hoping with some additional genetic data that Olga's going to generate that will have a different, address this question with some different kinds of data. But it looks like these populations are quite local, that they're not moving a lot between crops. Okay, so conclusion. The good news is we don't see evidence of highly insensitive isolates to either fludioxonil, so that's one of the components of switch, or to iprodione in Washington, and this is true for British Columbia as well. We have widespread insensitivity to elevate fenhexamide in raspberry. I want to continue to monitor these populations to see if these really are on the decline. And we need larger samples. We need to sample from the same field repeatedly, and we've started to do that. So we'll continue to do that. There's a clear correlation of fungicide use with the frequencies of insensitivity. So we're selecting in those fields that where we're using a lot of fungicides, where we're using a lot of one particular fungicide, we see a response to that use and selection for insensitivity. We have insensitivity to 3 of the 5 site-specific fungicides that are used to control botrytis. In some fields, in some locations, and it's not characteristic of county necessarily, there's some fields in Skagit that have high frequencies of insensitivity to all 3 of these fungicides. There are fields in Whatcom that have high frequencies of insensitivity to all 3. Okay, the frequencies of highly insensitive isolates that we had to iprodione in the late 1990s appear to have they're not around anymore. Probably declined due to fitness costs. This fungicide can be used again, but we need to be using it carefully. Otherwise, the same problem will repeat itself. If you use it intensively, we will select for high levels of insensitivity, but right now, it looks like it can be used again. We have large variation in these frequencies of insensitivity from field to field, and even from adjacent crops, adjacent plantings. So this suggests that these populations are quite local, and we'll have some other data hopefully in a year or so to confirm that, to address that. So it doesn't appear like we get a lot of long distance movement among fields. I think there's occasionally some movement. Some of the totally unsprayed fields that we've sampled, or unsprayed crops, things like the wild blackberries, we do see insensitive isolates occasionally. So there is some movement, I think, but by and large, the populations are quite local. That's what it looks like now. So one question I had is, so if we're seeing high frequencies of insensitivity to saprotanil and switch is still working, and I don't know, maybe that's something we can talk about later, is do we see a reduction in the efficacy of switch? Because one component in some of these fields does not, there appears to be high levels of insensitivity to that one component. So not to fludioxonil though. So is fludioxonil masking the insensitivity to saprotanil? Okay, some recommendations then. I think we should continue to not use Elevate and monitor the populations to see if they do decline in frequency. It's possible, I mean, this is pure speculation, but it's possible that it does decline. If they do decline over time, we might be able to use that fungicide again. We need to reduce selection pressure. So we need to reduce the number of sprays from each of those frac groups to a minimum. We need to rotate the use of them. We need to reduce the reliance on these materials. So we need to alternate. We need to mix with non-site-specific fungicides like Captan. This is a great way to reduce the selection for insensitive isolates. So we need to use other disease control measures where possible. Again, to reduce the reliance on fungicides as a control measure. The other aspect of this, and this is something else we're working on, is using these fungicides only when they're needed rather than on a calendar spray schedule. So trying to figure out when the fungus infects, how it infects, the conditions, the environmental conditions under which it infects, we hope will inform application timing, and that we can reduce the use, use them only when needed, and that way reduce the overall use and reduce the amount of selection that we're placing on these populations. So with that, I'll quit and take any questions you have.
Any questions? Please raise your hand and I'll track you down.
Looking at what you suggested at your fourth point there, only spray when needed. Would you suggest, kind of wondered this myself, you know a rain event's coming, would you suggest to spray right before the rain or immediately after? What do you think would be your best
so
bang?
the question is, so in terms of timing of fungicide application, whether it's best to spray before, if you can predict a rain event, and this disease is driven by wetness, wetness of the flowers. If you can predict those events, is it better to spray before or after? These materials are primarily protectants, so really they need to be on the plant before. Some of the site-specific fungicides, other than these, do have kickback, what they call kickback activity, so you can wait for an infection event and then use it after an infection event. But there's been no experimentation to support use of it in that way, so we think of these primarily as protectants. So needing to be on the plant before an infection event, if you can. The problem is predicting the infection event.
Wouldn't wouldn't it be best to get at least about 4 to 6 hours drying, whether it's before rain or after, but knowing that you're going to get 4 hours or 6 hours of drying, so it sticks to the plant. Yeah,
that would be wise. I would think. It's otherwise you risk it washing off, weathering. Yeah. So if you can get a period of dryness after the application, so that it does adhere to the plant.
Don't know if I'm comfortable talking to this group yet because I'm a newbie up here. But are you guys playing with PhD Frac 19 made by Arista? That's an alternative to Elevate.
Yes, Alan Schreiber, I think, can probably comment on that. He has been testing it, I believe. Whether you've been testing PhD, he's going to talk about it next. Yeah. Tom. Which Tom?
There are too many Toms here. Do any of these need buffering, or do you standardize these, buffer the water?
So you mean in terms of when we're testing them?
Yeah.
We don't. I mean, we do most of this routine testing is with formulated product, but we also follow that up. So when we find a certain insensitive type, we sample, like if we sample an insensitive type from Whatcom, from Skagit, and so forth. What we'll do is a more detailed test where we actually estimate the called EC50, so effective concentration that inhibits 50% of growth, but that involves many more concentrations and getting a much more accurate measure of the sensitivity. But for that, we'll use technical grade material. But we don't see a real difference between the response with the formulated, and the problem with formulated is we don't really know what else is in that formulation, but we don't use any buffers in the medium.
No, but in the field too, is there any effect of pH of the water?
I don't know. I can't really comment on that. I don't know. Does anyone else? So pH of the water, so your terms of like how well it sticks to the plant, or
well, I've always seen Captan in particular. It's very sensitive to the water pH. So I was just
that's not something I've experienced with. I don't know. Maybe Tom, you've been doing.
I don't know. I just heard anecdotally that acidification helps the efficacy of some fungicides, but I don't know which ones.
Yeah. Okay.
So I had a question. If all of the isolates are susceptible to the fludioxonilin switch, how can some arise that are resistant to the cyprodonyl switch?
If switch is actually controlling them, that's what you mean.
Yeah, because it's got both.
Right.
So any organism is getting simultaneously hit with both. So how is it that you can have isolates arise that are only resistant to one?
Yeah, that's a good question. Well, I mean, there's no evidence of any resistance to fludioxonil anywhere that I'm aware of. And I don't think, because the mechanisms are different, I don't think it would preclude if you selected, if it could become insensitive to cyprodonyl and still, right? I mean,
how does it survive the fludioxonil to become selected on by the cyprodonyl?
Yeah. Sorry? It inhibits it, definitely. Yeah. Is that what you mean? No, these are all, well, I don't know if, not too much is known about the mode of action, but. Right. Yeah, it's just not present in the population. Yeah.
It still won't keep down the resistant isolates.
Yeah. It could be due to just the dynamics of how much is actually, and that's a question that we've raised is how much is actually on the plant. It's a difficult question. It's why I use the term insensitivity until we're working on a fruit assay to try and get at that question. But it's how much is actually on the plant is it's a good question. So.
All right, and unfortunately we're out of time. Let's give a big round of applause to Tobin Peever.